So the next talk is by Felipe Llinares from Google Brain, who will tell us about learning to align protein sequences using language models.
Hello, everyone. It's a pleasure to be here. We just had a fantastic talk about how AlphaFold 2 has revolutionized protein structure prediction. And in this talk, I would like to take a step back, and I will be discussing how language models can improve homology detection and sequence alignment, which are techniques used to that techniques used to, for example, construct multiple sequence alignments that form AlphaFold's input. And also, this is the first iteration of this talk, so there may be some rough edges, and don't hesitate to interrupt and ask questions along the way. So DNA, RNA, and proteins are universal building blocks of life, from viruses to humans. And thanks to sequencing technologies, we now have access to tons of data. And this makes one wonder if borrowing ideas from NLP, deep learning could help decipher the language of DNA and proteins. There are plenty of potential applications, and many papers already began investigating this. The first mostly just applied the same techniques we use to model natural language to biological sequences instead, but others have gone beyond and started looking at specific downstream problems in the field. Again, AlphaFold 2 is without a doubt the most prominent and impactful application of transformer-based models to biological sequences, but there have been also many advances in other tasks like protein annotation or generative protein design, to name a few. However, applications of these models to tackle sequence alignment and, by extension, sequence comparison and search have received less attention. And the key challenge here is that, as we will see, sequence alignments are typically obtained by a discrete operation that, much like an Rmax or ranking, do not provide an informative gradient. And informally, what happens is that the gradient vanishes almost everywhere, and therefore models that use such a discrete operation as a layer cannot be easily trained end-to-end. And one of our goals in this domain was to overcome this challenge using differentiable optimization and kickstart what we hope will become a new generation of tools for searching and comparing biological sequences. So in today's talk, I will mostly talk about our first step in that journey. So this is a paper that will appear in Nature Methods this week, actually, but it's already available in bioRxiv freely. And this is joint work with my co-authors, Quentin Berthe, Mathieu Blondel, Olivier Teboul, and Jean-Philippe. And honestly, in many ways, I consider this to be more of a proof of concept than a finished product. We studied the problem of pairwise sequence alignment, which is a bit simpler than multiple sequence alignment, but it still shares most of its algorithmic backbone with the most popular sequence alignment tools like Jackhammer or HHBlitz, which were used, as far as I know, in AlphaFold, or also tools like Clustal Omega. So now our main goal here was basically to prove that transformer-based language models could bring drastic performance improvements in sequence alignment, which is arguably a field that has been mostly stagnant for decades. And I hope to have convinced at least some of you of that potential by the end of this talk. And I would like to organize this talk roughly as follows. So first, I will introduce the problem of pairwise sequence alignment in a mostly self-contained manner. I will explain why we care about sequence alignment in the first place, even if Michael already did a great job explaining that. I will describe how pairwise sequence alignment can be mathematically formulated and parameterized. I will introduce the Smith-Waterman algorithm and highlight some of the limitations of these methods. I will introduce our approach, so give an overview, and I will focus on our main methodological contribution, which is a differentiable version of the Smith-Waterman algorithm that we can use as a layer to train our models end-to-end. And then I will conclude with a brief discussion on how we train our model, show you some experimental results comparing to classical approaches in and out of distribution and some ablations, and then conclude with some related work and potential avenues for future work. So let's start with a quick introduction to pairwise sequence alignment. First, I would like to give you an intuition about what sequence alignment is trying to accomplish and why alignments are used as the input to many downstream tasks in computational biology. And I mean, we already saw most of what I'm going to say, but there were some questions, so I think it's still good to go over it a second time. With this in mind, basically, a fundamental question in biology is to determine if two proteins, like the two shown here in the slide, share some similarities in their sequence, beyond what we might expect by random chance, highlighted here, for example, in red and blue. And this is called homology, and it's caused by evolution from a common ancestor. Now, protein sequences evolve over time as the genes in our DNA that code for these proteins accumulate mutations. And roughly, these mutations can be substitutions, where one residue, like one letter in the sequence, changes to another. They can be deletions, where a part of a sequence is lost, or there can be insertions, where a certain subsequence is introduced. And informally, if we are given a pair of protein sequences, like here, we can think of pairwise sequence alignment as a rough attempt to infer the substitutions, insertions, and deletions that evolution introduced in each of these two sequences since their most recent common ancestor, which is a latent variable. We don't really know it. And why do we want to do this? Well, first, figuring out if two proteins are homologs or not can be very helpful, because homologous sequences tend to share properties of function. So if we have some new uncharacterized protein that we don't really understand, if we can find well-understood homologs of this sequence, is often the first step to annotate this protein. And in fact, how most of the available annotations in large databases like the UniProt knowledge base have been obtained in the first place. And secondly, the alignment itself can be informative, because inspecting which parts of the sequences are similar can give us some valuable clues of which parts of the sequences are most likely to be relevant to the biological function. For example, here in the slide, you can see a multiple sequence alignment for a set of real protein sequences. It's a PFAM domain. And something that you can see right away is that the entropy of every column is very different. Some columns are very, very low entropy. It's always the same residue. Others seem to kind of split the sequences into clusters. And then other columns just seem to vary wildly, as if they were noisy. And this is due to evolutionary conservation. Basically, conserved residues, those that have very low entropy, are very likely to play a key role in the protein's function. And then non-conserved residues are often not as relevant. Though, of course, this is just a rough guideline. Yeah.
So we're looking at alignments between proteins that are related out?
So they're basically related by evolution. So you can think about proteins evolving as basically as a tree branching from some recent ancestor. And ideally, we want to start. So if we have just one row, one sequence, we want to start finding the ones that are closest to you, which are the most similar. And then we go working backwards in the tree, like finding siblings that are farther and farther away. And at some point, we want to stop, because then these are so dissimilar that they don't add any useful information. But of course, aligning them is one part of the problem. The other problem is homology detection. Is if I have only one sequence, can you find me the sequences among a database of 200 million sequences? Which ones are the ones you should be aligning? There's these two sides, two aspects of the problem. Yeah. Mm-hmm.
In proteins, using proteomic data correctly.
Yeah.
So, like, are you integrating, fusing them to sequences, or you're doing real alignment and finding shared patterns?
Yeah. So it's more like, in this case, the problem would be I would have as input these two sequences, and now we need to figure out how are they related to each other. What is the best alignment you can find? And this is one toy example. It's not that these two are not real sequences, in fact. But basically, you try to find the most likely alignment between them that could be explained by evolution. And the reason why we try to do this mapping of one letter to another letter and potentially with gaps is because it's a model of how evolution works in real life, because of how DNA replicates. And in a way, yeah, you can view it as a way of data integration, because we have all these sequences that we don't know how they are related to each other, and we try to figure it out and put the puzzle pieces together, basically. Yeah, and then we already saw that another very important use case for alignments is covariation. So when we have these alignment matrices, we can find correlations between columns that are not necessarily close. And when this happens, it's often a clue that maybe those residues seem to be far in the sequence, but are actually close in the 3D structure. It's not always the case, as we just saw, but it's kind of a source of information that we want to exploit. And in the past, the state-of-the-art protein structure models use this in a very explicit way. I guess AlphaFold is much better in the way it has the potential to infer this information in a much more flexible way, but it's still likely relevant. And so in summary, sequence alignment allows us to infer if two proteins are homologs, that is, if they are evolutionarily related, and to understand which residues and which interactions between residues have been preserved by evolution. And then we can use this information to try to solve other tasks like protein annotation or protein structure prediction. So I will introduce some concepts now fairly quickly. So let's assume we have two sequences that we want to align. Let's call them X and Y. And they have potentially different lengths, N and M. And they belong to some finite vocabulary. And this vocabulary, it's much smaller than in NLP. For DNA, we will have only 4 letters. For proteins, we can have between 20 and 26. So it's very different in that sense. We will call our search space, which is just a set of all possible alignments between X and Y with a capital Pi letter. And we will use lowercase pi's to indicate like specific elements in this set, like specific candidate alignments. And we are going to be working with local alignment mostly, which means we actually want to align the best subsequence of X to the best subsequence of Y. And this is in contrast with global alignment that just tries to align all of X to all of Y. And perhaps the most important concept I want to introduce here is the scoring model, which, as the name suggests, is just a function that assigns a score to every alignment. And this scoring model has some parameters theta, which for now I'm just considering is some arbitrary function of the sequences we're trying to align. So it's in these parameters theta where the information about the sequences is encoded. But I will make more concrete what this is in the next slide. And basically, once we have a scoring model, the two things we want to know is, first, what is the optimal alignment? That is the alignment that has the largest score according to our scoring model. And then the actual optimal score, that is the score of this optimal alignment, is also useful because it's basically what we're going to be using if we want to classify whether two sequences are evolutionarily related or not, after some statistical corrections for confounding factors. But of course, the scoring model is not fixed. It's our job to design it, and this is the most important part, right? So roughly speaking, we want 2 requirements when we are thinking about what do we do for the scoring model. So first, it has to be predictive in the sense that we want that the biologically correct alignments are scored the highest, otherwise it's useless. And second, we want something that it's easy to optimize over. So we want something that lends itself to algorithms where we can find what the highest scoring alignment is. And because of this, what the field has converged to are additive scoring models, where basically we're just scoring every column independently of each other and adding some terms for gaps, which we will see later. And what this means from a statistical point of view is that we're kind of assuming that mutation, the likelihood of a mutation, for example, C turning into a P, is the same regardless of where in the sequence this mutation happens and in which sequence the mutation happens. And when we are willing to make this assumption, we can parameterize the model very easily. What we need is just something called often a substitution matrix, which is just a matrix that tells us how similar every pair of amino acids is, and a gap penalty function. So the substitution matrix, you can interpret it as a kind of reward, if you want to view it in the RL sense, that we get when we align one particular residue to another particular residue. And we have this for every possible pair of residues. And there are many, so these matrices, you can just find them available in the literature. Most often they are obtained by fitting some statistical model to small databases of known alignments. And in practice, we want to use a substitution matrix that was fit somehow in a database of sequences that is comparable to the one that we are going to align. So this is why, for example, when we look at a particular substitution matrix family, like Blossom, for example, we have Blossom 50 or 62 or 80. And we can think about this as a kind of hyperparameter. And then we have the gap penalties. By far, the most commonly used gap penalty function is the affine gap penalty function, which, as the name suggests, is just an affine function of the gap length, G. And here we just have two parameters to care about. The gap open penalty, which is kind of a price we pay for having a gap, regardless of how long the gap is. And the gap extend penalty, which is basically the slope. It's the cost we pay every time we make the gap 1 unit longer. Yeah.
Why is the diagonal not constant?
That's a very good question. The reason is because these amino acids have very wildly varying biochemical properties, and some are more special than others. For example, cysteines, the C, are very special, and they tend to form some kind of bond that stabilizes the structure a lot, and it's very hard to substitute. Like if you change a C by other letter, the protein often is broken. And you can see that this is reflected in, first you get a huge reward by conserving it, and everything else has a negative cost. But then there are other amino acids that are kind of like less unique. And there are other amino acids that even if it's not the same, it's similar enough that the protein is not going to break if you change it. And this is why sometimes we even have some positive values in the off-diagonal term. And yeah.
Yeah, I'm not sure I understood how you get the coefficients in the substitution matrix. Like the people who came up with this, like what are they trying to fit exactly?
Super simple. So you take a database of known alignments, which tends to be very small. And now you count frequencies of how often a specific pair of amino acids co-occurs in an alignment, and compute the log odds of basically P of A and B occurring aligned, divided by P of A and P of B. It's basically a point-wise mutual information estimate based on the alignment. And take the log odds and do some discretization, which, this was done in the 80s where floating point operations were too hard. So everything was discretized. And this is where it comes from. Yeah.
We could go, substitution can be done by just deletion and adding something, right?
Yes.
So does that mean that you need to have some kind of triangular inequality between R and gamma?
So yes, so in pairwise alignment, because we're just basically looking at two nodes, we ignore that problem. But then there is a very interesting problem, which is phylogenetic inference, which is iterate this over the full tree. And there, what you are saying is exactly right, and it's a big deal because, yeah, there are basically more than one path towards the same change, and there's, there are some family of algorithms that try to account for that, which is better but more mathematically involved, and other which, we just close our eyes and hope for the best. But yeah, you're exactly right. All right, so how do we actually compute an alignment? So the search space is very large, so good luck trying to do exhaustive search. But because we're using an additive scoring model, basically Bellman's principle of optimality applies, so we can just use dynamic programming. And I would just want to give you some high-level intuition about how we can actually implement this. So here we have an example with one particular alignment, and we can view them kind of like a path in a matrix that shows all possible pairings or possible pairings of residues in X to residues in Y. And here, for example, we start by matching the second residue in X to the third residue in Y. So we have this entry on. Then we have, again, two more matches. So we go down and right 2 times. Then we have a gap in X, which corresponds to moving horizontally 2 steps. And then, again, 2 more matches. So basically, every possible alignment, we can view it as a path like this. And hopefully, you can see that there are some constraints on which paths are allowed. Basically, if we're at any given point, we only have 3 possible actions, right? We can either move diagonally 1 unit, which is if we're actually aligning the next pair of residues. And then we can move 1 unit horizontally or 1 unit vertically if we introduce a gap in X or a gap in Y. And we can actually model this with a finite state machine, or which is where the hidden Markov models usually come from. And here we have a state for matches, which always has, like, all transitions into this state basically have the same reward, which is just evaluating the substitution matrix at the pair of residues that we are aligning. And the reason why we actually need this kind of diagram is because of the affine gap penalties, because we need to keep track when we have a gap, we need to keep track of whether it's the first gap or if we are extending the gap. Because then if it's the first gap, if we are transitioning from a match into one of these gap in X or gap in Y states, our penalty will be the gap open. But if we are just staying in the same state, it will be the gap extend. And we can basically take this to the extreme and kind of like unroll the finite state machine by creating one node for each of the states and every entry in the cell. So we basically, if the lengths are n and m, we end up with 3 times n times m nodes. And in this graph, some of the nodes it's a very sparse graph, and only that we have edges between states that can be, transitions that are allowed by our state diagram. And in this way, what we get is a directed cyclic graph. And we also introduce, this is more of a technicality, but we also introduce 2 states, a beginning state and a termination state. And these are here because we're doing local alignments, meaning we want to be able to not always start at 1, 1, and not always end at NM. So basically, we have these special states that kind of have shortcuts. So for any match state ij, like for example, this one, we can directly jump from the start. And we can also jump from any match state, we can jump into the termination state. So now we have this DAG. And what we can do is that because we are weighting the edges in the DAG by the rewards that we get by transitioning, it's quite easy to see that every possible alignment basically corresponds to a path between the beginning state and the termination state. Here I have shown one particular example, and you have one path with rewards in every edge. And basically, what we want to do now to find the optimal alignment is find the shortest path between B and T. And this is just one way to derive the Smith-Waterman algorithm. But basically, if we are thinking about this shortest path problem, we can just look at the diagrams of allowed transitions for every node type. And then it's very easy that if we have the value, the optimal score for each of these intermediate states, we just compute the maximum over all allowed transitions plus the particular reward that we get in the transition. And then we get a dynamic programming algorithm that basically works. We are filling value matrices starting at the top left and then working our way to the bottom right. And this is for pairwise sequence alignment, but basically methods like BLAST, which are just heuristics over this algorithm to make it faster, or Clustal for multiple sequence alignment is just based on recursively applying this to multiple MSAs. And methods like Hammer and H8Blitz, which are based on profiles, are basically the same algorithm, but with a more generative or probabilistic interpretation that basically makes Smith-Waterman be like Viterbi decoding. So it's basically the same in all cases. Now, these algorithms have evolved very little in the past decades, and this is kind of a testament to how useful they have been. But we are moving to a different era where we have way more data, and the age of these algorithms is starting to show as what before were edge cases now become more and more common. So in practice, how difficult it is to find homologs depends on how distantly related they are. And basically, the most interesting case now are sequences that have diverged for so long that their relationship is barely detectable. And we call this often the twilight zone of sequence similarity. And often we can only find these remote homology detection relationships if we have extra information, like for example, protein structures, which are more conserved. And here you have a real example of 2 proteins that we know are related. They are from the PFAM database. But actually, it's very hard because there are only 8 residues that are conserved. So if we just look at the sequences and all of the structures, it's quite hard. And if we apply the state-of-the-art pairwise aligners, they just completely break down. They don't get a single match correctly. And this is getting more and more common as our databases grow. And now we have about 230 million proteins in UniProt, for example, but 15% of them are completely uncharacterized. And this only gets worse if we look not at sequences, but at residues, where about 45% of residues have no annotations. And one of the reasons why this is the case is because it's quite hard to find homologs for many of these sequences. And this leaves us in the dark about their function and sometimes the structure if we cannot construct an MSA in the first place. So I would like to make a case that maybe using language models, we will be able to improve homology detection and sequence alignment and illuminate a bit this darkness in the proteome. So I would argue that one of the reasons why standard models perform poorly is that their scoring model cannot take context into account. So this is why we proposed our new model, which we call DEDAL. And the idea is super simple. Basically, we just want to make the scoring model depend on the context. So we are just going to put a transformer in here, make the transformer predict the substitution scores and gap penalties. We don't want to design it by hand. We just let the data speak by itself. So basically what we have is a transformer encoder TFI, which embeds our pair of sequences. Then the embeddings are fed to what we call a parameterizer, which just computes the substitution scores and gap penalties for this particular sequence. And then this is fed to the Smith-Waterman algorithm as before to compute an alignment. Now, what is tricky here is that we would like to be able to train this parameterizer and especially this transformer encoder end-to-end with a database of known alignments. But this is quite tricky because the Smith-Waterman algorithm, as I will explain later, is not differentiable. And it's also not that easy to implement in GPUs and TPUs in an efficient way. So for the transformer encoder, I think I will just skip it because we had an amazing talk yesterday. All I have to say is that this is completely standard. There is no novelty here whatsoever. So we just have an input sequence, and at the output, we have a sequence of embeddings, which are contextual, meaning, for example, the embedding for the second residue, G, depends not just on the value of G, but it also depends on the value of all other residues. And this is all we really want from the transformer. Then the parameterizer is also very simple. It's just a sequence of bilinear mappings of the embeddings. And this is the same for the substitution scores and the gap open and gap extend penalties. So it's kind of similar also to the outer product that AlphaFold uses, but in a much smaller scale. So for example, now the substitution score between residue I and residue J is just a bilinear map of the ith embedding of X, our kernel, and the jth embedding of Y. So nothing too fancy here either. But this is not useful if we cannot train the model. So first, let's see why it's tricky. So as I said before, we can view Smith-Waterman as finding basically finding a shortest path in a DAG. And every alignment that we can consider as a candidate is just one particular path between B and D in our DAG. And the weights are basically the parameters of the dynamic program that before were hard-coded, now it's the output of our transformer. Now, what we want to do is now find a representation of the alignments such that the score of every candidate alignment in our search space is a linear function of the Smith-Waterman parameters. And this sounds a bit convoluted, but it's actually super simple if we have an example. So for example, here we have 5 match states, like 1 at 2, 3, 3, 4, 4, 5, and then 5, 8, and 6, 9. So we can have a binary indicator matrix that just highlights that we were in a match state in those 5 cells. But we also opened a gap in 4, 6, which again, we indicate here, and we extended that same gap at position 4, 7, which we indicate here. So we can basically do this for every possible alignment. And then if we take the inner product between this type of representation and our parameters, we just get the basically the length of the path, which is also the score of the alignment. And what we want to do is just find the maximum over the set of all these binary indicator matrices of an inner product between our parameters and the representation. And this set is very large, but it's finite. So what we can do is take the convex hull. So basically every vertex is an alignment in this picture, and we can just complete the polyhedron by filling it in. And basically what we get when we do this is just that the Smith-Waterman algorithm is just a linear program in disguise. Yeah.
I'm a bit confused. You just had a transformer and then you moved to having this kind of weak mixes. So how was the transition done?
Yeah, so the transition is hidden in the parameters theta. So basically this theta have the substitution scores and the gap penalties. Before these were just based on this table. But now these are just an output from the transformer. These S, O, and E are now basically the output of these bilinear maps that depend on the embeddings.
So you feed the full pair of sequences into the transformer, and then almost like you a single triplet of matrices, and then
Yes.
you use it to align.
Yeah, so the transformer outputs embeddings. Oops. Then the embeddings are using a bilinear map to get these matrices S, O, and E. And then these appear here. And basically, all we want to do is to be able to backpropagate through the solution of this linear program. This is where I'm going, if that makes sense. Yeah, and basically, the problem is that now we want to backpropagate through the solution of this linear program, but the score is differentiable, but it's not smooth, which is already a bit problematic. And then the real problem is that also that the Rmax, so the alignment that optimizes this linear program, is piecewise constant. So basically, the gradient is going to be 0 almost everywhere. This is something you also get in things like ranking and so on. So what we did basically is to relax the linear program by embracing this interpretation that we have the convex hull of all possible solutions. So the effect of this relaxation is that at training time, not at inference, we're going to let alignments be in the interior of this polyhedron. And what this basically means is that we're allowing for fuzzy alignments, which are kind of like the expectation of some probability distribution over vertices. So for example, maybe it's 0.8 times this alignment and 0.1 times this one and 0.1 times this one. And we get some kind of like blurry alignment. And this is very similar if you're familiar with techniques like the Gamble softmax or the concrete distribution. This is kind of the same, but in the space of alignments, which is a more complicated, very high dimensional space with a lot of structure. And basically with this, we're going to follow two different techniques to relax the linear program. So that both are kind of exclusive. So we just tested both. And they honestly, we found them to perform very similarly. I will just describe them very quickly, just because I think they are much more generally applicable than to alignment. And in case some of you want to relax a linear program, maybe it will be of interest. But of course, we don't have a lot of time, so it will be super high-level glimpse. So the first technique is based on perturbations, and it's a paper that appeared in NeurIPS in 2020. And Quentin Berthet was the lead researcher here. And the key idea behind this paper is quite simple. The theory is not simple, but this is not our problem today. So that's good for us. And basically, all we do here is add noise to the parameters. So we have our linear program, but we are going to perturb it with some noise where the variance is controlled by a hyperparameter tau, which is kind of like the temperature of our relaxation. And now we define the perturb maximum as the expectation over this noise. And we can do the same for the maximizer. So we perturb the linear program. We look at what was the maximizer of this linear program, and we take the expectation over the noise. And this is actually very helpful because first, the perturb maximum is going to be smoother than before. So it's going to be smooth in a mathematical sense, which it wasn't before. And the perturb maximizer, now it's actually differentiable and the gradient doesn't vanish. So it's very helpful. And intuitively, you can see here a picture of what's going on. So of course, originally our linear program was deterministic. So, but now because we're injecting noise, this linear program is stochastic. So it will give us a different output depending on what the noise was. And this defines some distribution over the vertices. And this distribution is not tractable in general. We cannot know what it is, but we can always very easily draw samples from this distribution if we just solve the linear program with some perturb noise. And this is, in fact, a generalization of the Gamble max trick, if you may know about it. And yeah, basically, the most important thing for our application is that we can compute both the gradient of this perturb maximum and the gradient of the perturb maximizer with Monte Carlo approximations, which just using one sample tends to work pretty well. And I mean, I will not go into details of how we derive this, but for example, this is just very, very similar to the score function estimator in RL. And you can use this idea pretty much everywhere where you can write your problem as a linear program, like top K, ranking, optimal assignment, and all these kind of algorithms. And the second approach is based on regularization. And it was published in ICML in 2018, and it was led by Arthur Mensch and Mathieu Rondel. And as the title of the paper suggests, here they are trying to propose a framework to relax finite dynamic programs so that we can backpropagate through their solution. So if basically, very quickly, if we have some finite DP, we can always sort all of its states according to the order in which we would do the updates to solve the Bellman equations. And then again, we can kind of prove that this is equivalent to a sorted path problem, which we can write as a linear program. So if we wanted to smooth the dynamic program, something that we could do would be our one possible idea would be, okay, let's smooth the linear program instead. And one way to do it theoretically would be, let's add some regularization that intuitively this regularization is basically penalizing solutions that are very close to a vertex so that we are encouraging the model to get something closer to the interior. That is, and this will give us a smoothness. Now, this is nice in principle, but in most cases, it's very hard to solve this relaxed problem. So what the authors propose to do instead, as at first it feels like a heuristic, is let's not relax this, let's just relax the updates. And this defines some different solution. And like I said, it sounds very heuristic, but the authors were able to characterize how this behaves very rigorously and prove a lot of very interesting results. And one of which is the most relevant for us, is that for the particular case that the regularizer is the negative entropy, then actually both approaches are equivalent. But this is not the case for other regularizers, just for entropy. And also, we have efficient ways to compute the VGP and the HVP for both, basically the score and the optimal path. So either way, we can use perturbations or regularization. In both cases, what we are going to get are relaxed scores that are differentiable, smooth, and strongly convex, which is great. And now our alignments are themselves differentiable. So we are now finally free. If we have some database of like sequences under true alignment, we can fit any loss that we want and try to train this model by backpropagation. And I will go quite quickly over this. So that in practice, we use two specific losses, one for perturbations and another one for regularization that are kind of convenient mathematically. The first one is that so-called Frenchel-Young loss. I will not go into too much details. This is just, as the name suggests, is derived from the Frenchel-Young inequality. And it's a bit of a weird loss because we cannot really compute the value of the loss, but the gradients are super simple to estimate, and they are very convenient. And then the CRF loss, I think, is a bit easier to understand. We can write it just as a difference between the optimal score that we are predicting and the score of the true alignment. And for the particular case that we have entropy regularization, what we get is basically, it's just a complicated way to say we just have a negative log likelihood that is estimating the probability of, it's kind of like a softmax with the logits being the scores. And this means that what we are trying to train the model to do is to give a high score to the true alignment and a low score to the others, which is completely reasonable. And now, if you have not been lost, which you may have, something you may be thinking is, okay, wait a second. You just wasted 10 minutes of my life explaining how to make the alignments differentiable, but you're actually having losses that don't use the alignments, which is true. But it turns out empirically that if we don't relax the score, so if we set this temperature to 0 and we just try to backpropagate through these losses, the models are extremely unstable and they tend to get stuck in all kinds of plateaus. And when we started using this relaxation, these pathologies completely went away. So it was quite important. Yeah, of course.
So you have in your model a layer that actually solves that optimization problem.
Yes, and this layer, I will explain here. So it's just the Smith-Waterman problem algorithm. Same way to implement it, basically. Same updates. The only difference is what parameters you feed in. And actually, I didn't talk about it, but if you use this relaxation that I just explained, in theory, you could think that you need a different solution. But thanks to this paper that basically proves that you can just relax the updates, all that happens is that these updates that are a hard maximum become a soft maximum. And you can use exactly the same type of algorithm. So yeah, we have a layer that implements this Smith-Waterman algorithm. And the thing that is important for this layer is that if you want to do it in GPUs and especially TPUs, you don't want to implement it as a double for loop because it's going to be super slow, even if it's the most natural way. And you also don't want to use this type of fancy indexing, because if you think about the algorithm a bit deeply, and you want to say, okay, what is the maximum amount of parallelism we can have to implement these updates? The answer is anti-diagonals, right? So we can always compute in parallel the values for all elements in an anti-diagonal, but this is not very TPU friendly. So what we did, basically the whole point is we can do this rearranging of the tensors. And basically, this, especially if you're working with TPUs, which might not be relevant to everyone, but you can easily get almost 2 orders of magnitude speed up, at least based on our results. And this is something, if you're working with things like edit distance or dynamic time warping and all these kind of algorithms, they have the same kind of algorithmic foot very quickly. So the way we trained the model was with a multitask setting. So we had 3 tasks. One is just massive language modeling, nothing new here, BERT style. So for those who don't know about it, the task is just we take a protein, we mask out some of its residues, and the model has to predict, basically impute what were the missing values. This is mostly for pre-training. And then the 2 tasks that we're interested in were pairwise sequence alignment. So basically given 2 sequences that have a shared domain between them, possibly with some flanks that we need to discard, can you predict the right alignment? And homology detection, which again, we have a pair of sequences that have some domain inside, but now this domain may be either the same, in which case the sequences are homologous, or they can be different, in which case the sequences are not homologous. Yeah, so homologous means that the 2 sequences are evolutionarily related. So here we will be taking, for example, 2 rows. So this you can think about it as 1 row of a multiple sequence alignment, and this one would be another row of the same multiple sequence alignment. Whereas here, it would also be rows from a multiple sequence alignment, but different ones. So it's kind of like, can you tell me if these 2 rows are from the same MSA or from a different MSA? This is kind of the task. Yeah, again, I will pretty much skip this, but so for massive language modeling, we just use the UniRef50 database, and then for the sequence alignment and homology detection tasks, we use a database called NASSMA. It has around 20,000 manually created multiple sequence alignments, covering about 1 million sequences, also not a lot of data. And we can use this data to perform inputs both for the pairwise sequence alignment task by taking pairs of sequences in the same family, and also for the homology detection task by taking pairs of sequences in a super unit, which is called a clan, which is just a collection of families that are evolutionarily related, but with a very distant relationship. And in this way, we have kind of a bi-level, like 2 levels of difficulty, because if the sequences are in the same family, it's a bit easier. And if they are in the same clan, but in different families, detecting homology is a lot harder. So how well does this work? So if we go back to this motivating example that I had at the beginning, it's quite striking that even if the baselines were pretty much unable to get a single prediction right, the deep learning based model only makes 4 mistakes. And these 4 mistakes are all within 1 position of the ground truth. So it's almost a perfect alignment. And I think I will skip this in the interest of time, but we can just interpret the scores is all I wanted to say here. And I wanted to show some very brief quantitative evaluation, not just a cherry-picked example. And I think the most important point I would like to emphasize here is I think we should really always look at out-of-distribution performance in this type of work. And the way we did this was by splitting our data at the clan level. So we basically left out 1,000 clans and used those for an out-of-distribution split. And in this way, it's kind of like having classes at test time that our model never saw at training time. And of course, we also show in-distribution performance, which is more like how well do you generalize to new sequences, but from classes that you know. And this is not so important. Basically, we created 2 variants of the task by adding flanks to make it a bit harder or not. All right. And as for baselines, we chose the best performing substitution matrix and cap open and cap extend combination basically out of like 1,000, almost 500 experiments. So it was pretty exhaustive. And we're going to show the F1 score as a metric for alignments, which is just a geometric mean of precision and recall. And then for homology detection, we will show the AU-ROC. And nothing too special about hyperparameters. It's a pretty small model with nothing fancy. The only slightly weird thing is that we use sort of a curriculum strategy where we introduce tasks one by one. And this improved performance. So how well does it work? So let's just start with alignment performance in the out-of-distribution split, so kind of the hardest setting. And this was already super encouraging because something I should say is, so here we are stratifying performance by the percent identity of the sequence pair that we are trying to align. All this means, so percent identity is kind of how similar the sequences being aligned are. And the highest, the more similar, and the way the task becomes way easier if the percent identity is high. And basically remote homologs, so these very faint relationships are over here in the plot, are the small, very small percent identities. And these are the ones we really wanted to do well at. And actually, despite being out of distribution, it was already very encouraging because we can almost double the performance in the most challenging bucket. And we get also a quite sizable improvement in the second most challenging bucket. And if we go to homology detection, the results are even better in a way, because again, for the most challenging bucket, when we are looking at sequence pairs that are in the same family, we get a pretty large boost from AU-ROC of 0.7 in the baseline to as high as 0.9. But the one we found the most interesting is that in the problem of predicting if two sequences from different families but in the same clan are related or not, which is often not really done with sequence alignment. That is not pairwise sequence alignment. You either use profiles or very often structures. And indeed, the baselines are barely doing random guessing here. We found that just by using the pair of sequences, actually, we had a pretty non-trivial AU-ROC of about 0.72. So this was quite nice. And then, if we go to the in-distribution split, it's not really interesting because the deep learning models here, I mean, there is no contest anyway. So the deep learning models in distribution, they are extremely good at interpolating between known data. So here we are multiplying performance by 3 in the most challenging bucket. And if we look at homology detection here, the model just basically has no problem detecting, even in this super challenging case, it makes almost no mistakes. And I think, I mean, I don't want to send the wrong message. I think these results are good, but I think in distribution is always a bit misleading because it's a much easier task to detect that a certain sequence has a domain of a type that you already know, which is what we are doing here, than in the previous out of distribution slide, where it's like, I give you 2 sequences that are mostly unlike anything you have seen before. Can you detect that these two are actually similar when the similarity is very weak? This is way harder and I think more relevant. And okay, I think because we are already over time, I mean, this is not super important. So I, of course, would like to mention some related work because it's good practice. And I think the most relevant of these 4 is this paper here. I think in all fairness, we have to call attention because they basically concurrently developed a very similar technique to what we did. And it's complementary in some sense because their encoder is simpler because they just use a single layer CNN. So their alignment results are less strong. But they do some super cool stuff like backpropagating through AlphaFold, like connecting the alignment atop the whole AlphaFold stack and trying to optimize end-to-end. And I think this was a super cool paper. And we also did some work where we repurposed all this differentiable alignment machinery to help in a paper that is trying to improve genomic sequencing, basically, in long grids. Yeah, I think I'm over time, so. Yeah, so just to conclude, I think the whole general field of artificial intelligence and in particular language models and transformer-like architectures applied to proteins and also DNA, I think will be extremely exciting in the following years. On the alignment side of things, I think extending this work to multiple sequence alignments and profile alignments will be fairly easy, because the algorithmic backbone, like I said, is basically the same. And the most challenging thing to be solved would be how do we encode not a sequence, but an MSA? And this problem has already been solved by the AlphaFold folks. And also, for example, in Facebook AI Research, they have MSA transformer. So we could just pretty much change our encoder and retrain the model with MSAs. And something that I think is also very interesting is structural alignments. So before we had a brief mention to fault-seek, and the way fault-seek works is by discretizing the structure with a VQ, VAE, and basically getting a sequence where your symbols are not residues, but some categorical variables that encode local information about the structure. And then they do a pairwise sequence alignment on these latent symbols. So we could, of course, do something like fault-seek, where instead our inputs are to structures. We have encoders that encode these structures, and we learn everything end-to-end. And more generally, I think there are other fields like protein annotation, especially protein design with generative models, that are going to be super exciting in the next couple of years. So yeah, I'm a bit over time. So with this, I would like to conclude, and thank you all for your attention.
Thank you very much for your presentation. So I have a question about the loss function. The choice of
Yeah.
So my question, why did you choose this function?
So the honest answer is we chose this loss function because the way the gradient that you get, it's very easy to compute. And it's just, the gradient is just the difference between basically the alignment you predict and the true alignment. In fact, you get the same gradient. Maybe let me explain it this way. You get the same gradient if you just put L2 loss on these binary indicator matrices. And we talk about differential-Young loss and not this L2 loss because it's more theoretically principled. I mean, there are strong connections with the with the perturbation methods that are just theoretical issues. It's not super relevant in practice. But yeah, the honest answer is computational convenience, because we don't need to backpropagate through the alignments. And then the backward pass gets way cheaper. And it works well. So we basically tried the cheapest thing we could do, and it was already working. So we didn't want to make something more complicated.
And the minimum of this function, is it unique, or we can do just not minimum?
No, so this function, this is because of the Frenzel-Young inequality. Basically, you can prove, it's a bit of a weird loss, but you can prove that the loss is always greater than 0, even if it's not at all obvious. And it's only equal to 0 when the predicted alignment and the true alignment are the same, which is a property we want in a loss. But to prove this, you need the Frenzel-Young inequality. This is why it's called this way. Yeah.
Do you think that this approach will replace the existing tools such as
That's my hope, but the honest answer is I think there is a bit of work left, and the work is in 2 directions. So one, I mean, we just need to bridge the gap to multiple sequence alignment and profiles, which I think I'm fairly confident is not going to be a problem. The other bridge to be, the other gap that we need to bridge is the scalability. Like this is still slower than BLAST, and BLAST is already not too fast. I mean, for our current standards. So I think this is the area. If we want to make this truly, truly useful, we need to be faster. And this is what we're trying to do now, basically. Yeah.
I'm still a bit confused about the intuition, to be honest. Like, you say that what was before, the problem was that it's like simplistic and it assumes that everything is independent. So instead, you look at the whole sequence and then predict this sort of the same independent sequence, right? But like, what is the intuition here? Like, why looking at the sequence and like making this kind of same simple setup works and helps?
Yeah, I see. So the intuition I would give is kind of like, think about it about conditional independence, right? So before we were in the previous model, I mean, these additive scoring models, you're assuming this independence across positions right from the start. Now we are still assuming this independence across positions, but it's conditional on the embeddings. But the embeddings already have this context sensitivity thanks to the transformer. So it's kind of like cheating in the same way as, for example, you can have an encoder-decoder model that is conditioned on the embeddings in the encoder, and you have this sensitivity to the full context in the sequence. It's something similar. So this is why the whole thing kind of works. It's because the transformer is so flexible that it can aggregate all the information it needs in the embeddings, so that the very simple, very limited Smith-Waterman still gives you a lot of flexibility. That's all intuition I can give. Of course, I think it would be better if we had the actual dynamic program to have a memory, but then you lose optimality. Like you cannot, or at least I'm not aware of any way to do it. But this is also an interesting thought, which is what if we close our eyes even more and we just trust the transformer to do the right thing? I mean, we're doing it with code all the time, right? It learns the structure of the code. Maybe it can learn the structure of alignments completely unconstrained. And we tried it. It works worse out of distribution, but we didn't try to scale it up really large. So it's interesting. Maybe it will be better. Yeah.
So from the problem context, do you have an assumption on the length of computations and corruptions? The hyphens.
Ah, yes. So you mean for this task here?
Yeah. Do you have like a maximum length or?
Yes. So for computational reasons, we are working with sequences of length. I think it was 1023. I mean, we want 1024, but we have one end of sequence token. So.
Talking about where the corruption can appear.
Yes, the corruption. The corruption is random, and it's on 15% of positions, which we didn't try to optimize. I think.
Because you showed a baseline which computed a very long corrupted part of the sequence.
Mm-hmm.
So if the baseline presuppose or assume like a length, a maximum length, a small length for some probabilistic distribution, it wouldn't be very bad as you showed. The length of the corruption of the imputation.
But so one thing I didn't. So the corruption is not really when it comes to the alignments. The corruption is not present. This is just a different task. It's just a task we use for pre-training. But when we are trying to do the alignments, the sequences don't have any corruption, neither for our method nor for the baselines.
Yeah, but you're right there.
Ah, okay. Okay, I think I know what you mean. So you are talking about these flanks? Yeah, okay.
When you show the baseline with all the other, we compared a lot of methods. In the baseline, the solution that the baseline found was with a lot of corrections, and there is some alignment.
Yeah, I'm trying to understand. So you mean here?
Yeah.
Yes.
You see
yes.
One. So the Mara one is not very bad. It's not very good, I think. Right?
Yeah, it's quite off.
It inserted a lot of corruptions.
Yeah. So what really happened here is that you see these two Cs that are aligned. So as I said before, the cysteines, the Cs are super conserved. So what happened in this particular failure case is that the classical models got misled and kind of anchored the alignment on these Cs, and then it tried to make it fit afterwards. But this is wrong because these are not the ones that should be aligned. There are 4 cysteines in this sequence and another 4 cysteines here. And this one basically got confused about which of the two should be paired. And then everything went downhill from there. This is why you have this huge bug here.
If from the beginning we had an assumption like we can only have a corruption of length 2 or 3 or 4, then we would
yes.
have a better result.
Yeah, you can add, but the problem is then you lose the flexibility because in some cases you might want this kind of constraint like you were saying, and we can actually do it. You're totally right in the dynamic program. But then sometimes you have alignments that have actually a really long gap in between, and then this doesn't work anymore. And we would want to cover both cases. But yeah, thanks. I mean, we can also talk after if you want.
Okay, so in the interest of time, maybe
let's
end here. Let's thank Felipe again.
Thanks.